Olson Lab focuses on exploring and understanding neuroplasticity, or the induced growth of neurons. In 2018, our lab published a paper on how psychedelics, such as DOI, DMT, and LSD, were able to induce growth in neurons, potentially opening an avenue through which we could treat the neuronal degradation of neuropsychiatric disorders, such as PTSD, substance use, and depression, to name a few. Now, the drawback of psychedelics is that they induce hallucinations, which can cause harm in people who have the very neuropsychiatric disorders that these compounds could treat. We have therefore made it a goal to find safer and more efficacious ways in which to induce neuroplasticity. Since then, many studies have delved into how psychedelics work, but the mechanism is incredibly complex and not fully understood. What researchers seem to agree on, though, is that these mechanisms are mediated by the serotonin 2A receptor. To better understand this mechanism of action and to potentially differentiate the pathways that cause hallucinations and neuroplasticity, we wanted to develop tools that could illuminate the various receptors or pathways involved in this process. We aim to make clickable psychedelics, termed clicks, with an alkyne embedded in their structure that would allow them to be labeled with a fluorescent fluorophore so that we could image and visualize how these compounds move throughout the brain, which regions they target, and which receptors they interact with. We began by adding an alkyne to the most chemically accessible positions on serotonin, which is the endogenous ligand of this key 5-HT2A receptor, as well as 5-MeO-DMT for its similar structure and binding profile, and we expanded this library of compounds to LSD as well, as it's a psychedelic with many receptor targets and could help show what these chemical tools were capable of. We synthesized these clicks and evaluated them in biological assays to compare their activity to the parent compound. We found that they had similar hallucinogenic potential and receptor affinities, so they should act the same in these biological systems. We then showed that these compounds could bind to the target receptors even in cells, and once we added the fluorophore, shown here in magenta, we could tag the alkyne of these compounds and reveal their location. To take this a step further and actually track a psychedelic as it moved through the brain, we gave the LSD click to mice, observed the characteristic head twitch that indicates hallucinogenic activity. Then we added a fluorophore, and for the first time, we could visualize where the psychedelic compounds spread in the brain. This gave us a lot of information. For example, we found that the higher doses spread farther throughout the brain and caused hallucinogenic-like phenotypes in mice, while lower doses of the same compound didn't reach as many receptors and didn't cause hallucinogenic activity. We're following up on these studies with other animal models and different assays to better understand where hallucinogenic compounds can spread and which receptors they can label and activate. But we have made a huge step towards understanding psychedelic movement in the brain, and we hope this will help tune our compounds to better target receptors of interest for neuroplasticity while helping to eliminate the hallucinogenic effects.